All right, so we just went over how a protein is made. So remember, we go read the code, bring the code to the ribosome, attach the building blocks. Voila, protein. All right, so the first step that we had was that the DNA was getting translated, we call it translated, into RNA. And the RNA is not making an exact copy. It's going to make what we call a complementary copy. So it's going to use the base pairing rules, and it's going to match the bases that it can use. So for example, the RNA right here would match an A in its copy, in its complementary copy. So what I want you to do, I'm going to give you about 30 seconds to, on scratch paper please, or your little whiteboard or something that you can write on, write down what bases would match here. And remember, RNA can't use base T. So if you're trying to use a base T, you got to change it to the U. All right, so what you should have is this. So AUG, GGC, CUA, AUG, UUU. All right, so a couple things, remember, I don't have a T here because RNA can't use that T. You'll also notice I've grouped these into threes. That's gonna make our life easier. When I do these practice problems, when I'm writing them out, I actually draw lines between every three bases because the DNA is read in groups of three. So it just kind of visually makes my life easier. Here I've put a space so you can see where the groups of three are. All right, so this RNA is what's going to go to the ribosome. The ribosome is going to read this and use this code to attach the amino acid, which is the building block to make the protein. So this is the code we need to be able to read now to know which amino acid or which building block we're going to attach. So if I read this and I'm building Legos, I'm like, okay, that's my blue Lego. Every time I see AUG, I know I need a blue Lego. Your cells are doing the same thing. They're like, okay, every time I see AUG, I need this building block for this protein. So we go to our ribosome. This is a really complex looking chart. I don't want you to stress about it though, because I'm going to show you how to read it. So this is called a codon chart. What's a codon? These are codons. The groups of three are called codons, okay? So again, we're gonna read this RNA in groups of three. All right, so what you'll notice in this codon chart is it says first base, second base, third base. There's A's, there's G's, there's U's, there's C's, all over the place. So how do I read this? Well, the first thing I need to know is what is my first base in codon? So if I go back, my first base is an A, okay? So that means my answer is somewhere in this A column, okay? My second letter is U. So that means if I look at here, it's in the U column. So I know it's somewhere in here, but I already know from it being A that it's somewhere up here, okay? So A is these four boxes at the top. U is right here. So we know it's somewhere in those four. So to figure out which one of those four boxes it is, now I need my third letter, which is G. So there's G. So G leads us to methionine. So we got A, U, G, methionine. Now, underneath your codon, so you should have that on your scratch paper still, write this amino acid. Now, you don't have to write the whole thing. We use abbreviations because there's a lot of letters in these things. So use the abbreviation of the first three letters. So MET are the first three letters for this one. All right, so the next codon we have is GGC. Let's see if we can find where GGC is. So take a moment, see if you can find where first base G, second base G, third base C is. And then write it on your scratch paper. Okay, so first base G, we know it's somewhere in this row. Second base G, okay, we intersect here. Third base C, so we know it's this box right here, so glycine, and again, we abbreviate GLY. Okay, on your scratch paper, you have CUA as your next code, or your next codon. So see if you can find where CUA is. Okay, so we got first letter C, so we know it's down here somewhere. Second letter U, so again, we know it's right here. Some of you have started to realize that the third letter doesn't always matter, because, for example, we've got 4 of the same things right here. That's actually a really cool thing that helps the DNA if it makes mistakes. So we've got C, U, and then A. So leucine, our abbreviation is LEU. Okay, your next one is AUG again, so it's the exact same AUG, AUG, so MET, and then U, first letter, second letter U, third letter U, phenylalanine, so abbreviation PHE. And if you're struggling a little bit, don't worry, I'm going to give you more practice. Plenty of more practice, in fact. All right, so these were the amino acids that we ended up with: MET, GLY, LU, MET, and phenylalanine. All right, a few things. Like I said, just write the first 3 letters of the amino acids. Otherwise, it takes forever to write the full things. They're huge. There's a few exceptions, and you might want to jot these down. So asparagine, glutamine, and isoleucine, and I'll show you why. So if I go back, here's asparagine. It's ASN. The reason it's not ASP is because there's another amino acid right here, aspartic acid, that starts with ASP, so that one gets to be ASP. So this one's ASN. Isoleucine, I don't know. Here it is. I don't know why it's not ISO. Occasionally you'll see ISO, but you see ILE as its main abbreviation. Okay, and then our last one is glutamine and glutamic acid. So you'll notice glutamic acid, glutamic acid, GLU, can't do that. So they have the same first 3 letters, so glutamine gets to be GLN. All right, so all proteins, what's really cool is they start with AUG. Now, your chart on the one that I'm giving you in your notebook is going to have a word there. Okay, so you'll notice mine doesn't actually have a word, but yours will have a word here as well. It has the word start. And you'll notice right here, 1, 2, 3, there's 3 words that say stop. What's really cool is these are the way that the ribosome knows to start and stop proteins. Okay, MET will often occur in the protein as well. So while every protein starts with MET, it can also just occur in the general protein as well, which is why you don't see the word start written on my chart on the screen here. But on a lot of them, they'll say MET and start. The stop codons, they never appear until the end of the protein. So as soon as you get there, the ribosome's like, oh, done with the protein, and stop. Right, so again, MET is the start protein, can also be found in the protein as well, and there are amino acids that stop the protein, okay, and I just pointed those out to you.